Hi everyone, good morning and or good afternoon, depending on where you are in the country or where you are in the world. Welcome to week 6 of your journey into the world of the unseen. Now, the big takeaway for this week is that bacteria are not the only bad players in this game called life. There are a myriad, a bunch of eukaryotic pathogens that we will examine, or that we do examine in our study of microbiology. And while many of those eukaryotic pathogens are microscopic, like amoeba, paramecia, um, some are actually large enough to be seen with the unaided eye, including some species of fungi and some protists. Um, we even study tapeworms and roundworms and pinworms in microbiology, and those are certainly large enough to be seen with a naked eye. I Somewhere along the way in the past, those organisms got um, linked in with microbial pathogens, and I think that's because of the fact that they cause disease. I'm not sure, but that's sort of the historic um, program. And so, in this week, in this unit, we will talk about, or you will examine tapeworms and pinworms and those kinds of things. All right. So you will recall, most of you from general biology, like in high school, maybe, that bacteria are prokaryotes, and this is something you've heard me say before. These are organisms without a nucleus, while eukaryotes, remember the EU means true, so these are organisms with a true nucleus. Um, and there are unicellular eukaryotes, and there are unicellular eukaryotic pathogens, which is what one of the things you'll be looking at this week. And so this week we kind of bridge the gap between prokaryotes and eukaryotic pathogens by doing comparative surveys of each. Now, I suggest that as you progress through your surveys, that you look for similarities between pathogens, signs and symptoms of infection, and treatment protocols as well. So I've provided links to two short videos to help you understand the concepts here in topic 6. Now, the first describes the basic procedure and purpose of ELISA. That's an acronym, stands for Enzyme Linked Immunosorbent Assay. And it's a way to determine if someone has been exposed to some whatever pathogenic microorganism. In Connect, you will see an image, a photographic image of the results of an ELISA test. And one of your assignments this week is to use that ELISA test results, and then you're going to answer questions about it like that. It asks you, tells you to sketch that test result on your page. You do not have to sketch that. You can just copy that image. You'll see it's like little tubes. Some of them have blue and some of them are clear. You can just copy that image, paste it onto your assignment, onto your submission uh, paper form, and then just answer the questions based on that. Save it as a PDF and upload it. Okay, you do not have to sketch that. In the other two assignments, you must sketch. You must hand sketch. You can do this. Some of you have the ability. You have the little digital tablet in the pen stylus, and you can actually do a sketch. Some of you may have to, other students in the past have sketched on paper, take a photograph with your phone, take a photograph of that, upload it into your assignment, your submission. You can do it like that. If you're able to sketch download a hard copy, sketch it on the paper, and scan that and upload it. That way you can do it that way. So there's lots of different ways that you can do this, but you must hand sketch it. Okay, and that's on your prokaryotic pathogens and your eukaryotic pathogens. Okay, so just remember you must sketch those, and you will lose credit if you do not sketch those. Okay, so and then the other let's see, the ELISA, you'll learn about the immunology lab. And the second video is a brief introduction to eukaryotic microorganisms. Um, be pretty good to watch that before doing the homework exercise. Housekeeping items for the week, working on topic 6, that's microorganisms and humans. You want to make your initial discussion post no later than Friday, February 20, 11:59 p.m. GCU time. And remember that you must make a minimum of two peer responses over 3 different days so total of 6, in order to earn full participation credit. There's a pre-lab questions assignment for labs 8, 12, and 13. That's due no later than Sunday, February 22. And then you have the 3 labs to do. Okay, these are all through Halo, not through Connect. Um, all of them are due no later than February 22. That's Sunday coming up. So the immunology lab that uses the ELISA result from Connect to complete the worksheet. The ELISA result is in Connect. The assignment that you're going to submit is in Halo. Copy that image, paste it into the assignment on Halo, answer the questions, save it as a PDF, upload it in Halo. The infectious diseases lab or exercise, you need to describe the morphology. That means morphology, that shape, that means you sketch it out. Identify a disease, describe the treatment for each organism, and you're going to submit that. And then that's lab 12, and then 13, the eukaryotic pathogens lab, you're going to describe again the morphology, sketch it out as best you can. This is not an art class. Answer the follow-up questions and submit the worksheet. There's also a matching section down below at the bottom of that sheet. Save everything as a PDF and then upload it into Halo. All of those are due no later than Sunday. Remember that your benchmark assessment, remember the lab report that some of you have submitted already, but should have started last week. It is also due no later than Sunday, February 22nd. There's still time if you have questions um, about identifying your unknown. I posted, and I told you this last week in my announcement, but I posted a template for your lab report in class resources. And I also posted a student sample, an example for you to follow to kind of get the idea of how to put your lab report together. So there should not be any questions on the format. Your results, the images that you find in your unknown file. So each of you has an unknown assigned through Connect, 1 through 10. You get 4, 5, or 6 images of results there that have been tested and been completed on your behalf by a lab assistant, for instance. All you need to do is you want to copy those images, of course, paste them into your lab report, but then you need to do a brief description of each of those and look at the student sample report to show you how that table is put together. And then you interpret the results of those tests to identify your unknown, number 1, number 2, number 7, whatever, okay? On the right side of the topic page, topic 5 page, you'll find a vertical menu. Down near the bottom, it says Identification of Unknown Bacteria, Lab 14. If you click on that blue link, that will open up the manual for this assignment if you didn't find that. And it tells you in there how to interpret the results of your tests. And then in figure 7, I believe it's figure 7, there's a dichotomous key that allows you to trace based on the results of your test, trace down, you got gram stain, positive or negative, positive over here, and you go down. Um, Coagulase test, positive or negative. Positive, you go over here, negative, you go over there. You just follow it down step by step until at the very bottom you can identify your unknown. It's E. coli or Streptococcus aureus or whatever. Okay, that's how you use that dichotomous key um, with the results of your test, and then you're going to follow the format and write up your report that way. As far as materials go, just list the materials uh, just kind of generically. All the materials you're going to use for all of the tests. You don't have to break it down per test. That's under materials. Under methods, if your results showed a gram stain, you want to do the 4 or 5 steps to do a gram stain. You don't have to go through the whole thing. I used um, a clean environment. I put on gloves. I put on a mat. You don't have to go through all that. Just give me the basic steps of the gram stain, the basic steps of the macaque auger, the basic steps of the coagulase test, like that. That's under methods. And then, of course, self-explanatory, I think, results, discussion, and conclusion. You need at least 3 references, authoritative references, properly formatted, and make sure you put your references on a page all of their own. Everything else can be sort of continuous and connected. When it comes to references, you want a separate page at the very end. You want to label it references in the center, that in bold, and then you want to present your 3 references, preferably hanging indent. Make sure you skip a line between them. Okay, you need at least 3 there. All right. Oh, heads up, next week, final exam. It is comprehensive. Check out my video review, midterm review, and I will post a list of topics for you from weeks 4, 5, and 6. All right. Questions, issues, problems, contact me as soon as possible. We're getting to the end. Next week is our last week. So contact me as soon as possible. Message 804-229-7993. You can use your Halo inbox or message box. 2 quickest ways to get a hold of me, and certainly you can use your campus email, jonathan.jeffries@my.gcu.edu. You know how that goes. If you're using message, make sure you give me your first and last name and also the class that you're in. This is Microbiology 205 Lab. 205, yes, 205 lab. I teach both lecture and lab. So make sure you tell me that you're in lecture or you're in lab. Okay, that's all I have for you. Make sure you get that benchmark assessment in by Sunday. And that's all I have for you. Have a great rest of the week.